Hello everyone, my name is Marola Isa and I'm a first-year PhD student at Case Western Reserve University working with Dr. Chris Worth as my advisor. Today I will be presenting our work on active particles, most specifically the influence of nanoparticles on the dynamics and clustering of active colloids proximate to a boundary. So what does it mean for a particle to be active? In order to help understand active motion, first, it's important to understand what passive motion means. So passive or Brownian motion is a randomized motion which is experienced by microscopic particles suspended in a fluid due to thermal agitation. And this motion is depicted by the first video on the left. And a particle becomes active when it has the ability to propel itself in the media in addition to undergoing Brownian motion. Generally, propulsion can be generated by chemical reactions or light or electric fields. However, the active particles we work with in our lab move due to a chemical reaction via a process called self-diffuse euphoresis, which means movement due to concentration gradient. So our particles are made out of polystyrene and platinum, and when hydrogen peroxide is added, what happens is that platinum catalyzes hydrogen peroxide and generates phoretic effects, or gradients in the concentration and charge that renders those particles active. This is also depicted in the schematic in the second video. What I would like for you to note from this slide is looking at the passive and active path trajectories, so those two figures, is the difference in the trajectory length for those two. This is important because being able to fabricate active micromotors that are capable of supplying mechanical work could be very useful in a wide range of applications, such as bioimaging and sensing, targeted drug delivery, and also for modeling biological phenomena, such as bacterial colonies and interfaces. And the work I'm presenting today represents active colloids as models for biological systems. So beginning with the Janus particle fabrication, first we form monolayers of polystyrene spheres that are dispersed onto silicon wafers via spin coating technology. Then the wafers undergo physical vapor deposition, which allows for depositing platinum onto those particles. And the third and final step involves retrieval of the Janus particles, which is done by sonication and washing of the suspension. And lastly, the particles are redispersed into ultra-pure water. This image on the lower right represents the inside of a physical vapor deposition machine. The platinum pellets sit in a crucible bowl, which is located here, and an electron beam hits the pellets and allows them to evaporate upwards, considering this process is taking place in vacuum. Meanwhile, the silicon wafers housing the polystyrene monolayers sit at the top, which is not shown, but it's here, and are facing downwards parallel to the ground, allowing for the particles to be partially coated with platinum. So a quick overview of our experimental setup. We install the fluid cells onto microscope slides using a secure seal spacer and a cover slip on top in order to minimize evaporation. In our experiments, we utilized light microscope and a CCD camera to capture all videos instantly after assembling the fluid cells. Also, the images were collected and analyzed at a frame rate of up to 16 frames per second. And after the videos were recorded, we processed them using Fiji and then tracked the particle trajectories. Note, the image of the microscope slide shown was captured well after an experiment was done. As you can see, there's a large accumulation of gas in the fluid cells. This doesn't happen during video capture to the extent that is shown in this picture, and thus doesn't really affect our observations. However, I wanted to display this image to emphasize the time-sensitive nature of our experiments. Moving on to data analysis and methodology. So in our initial work, Janus particle concentration was set at infinite dilution in order to minimize the effects of surface interactions that could arise between nearby particles. In order to determine the level of activity, which is represented by the particle's propulsion speed, we processed our captured video and were able to generate the micrographs that are seen in step 1 for non-active, which is image 1, versus active colloid, which is image 2. Then we analyzed the mean square displacements, or MSD, for all captured particles. Note, MSD represents the square difference in position of particle I at time t in the initial position of that particle averaged over many particles. And the two equations that you see displayed at the bottom are the MSD equations for passive or Brownian motion, which is equation 1, and active motion, which is equation 2. The 4 in those two equations is a coefficient relating to the dimension, which in this case is 2d. The proportionality constant d naught is the diffusion coefficient, tau is lag time, and Vp is the apparent propulsion velocity. Also, this is what a typical MSD plot looks like for non-active, which is a black line, versus active and 5% fuel, which is the red line. What I would like for you to know is the slope of those two lines. You can see how the red line has a much steeper slope compared with the black. And the slope is nothing but the diffusion coefficient for the colloidal particles, which is a direct indicator of particle activity. Analyzing our data after varying the hydrogen peroxide concentration in the suspension between 0.1 and 10%, we observed that the propulsion speed increases roughly linearly with increased fuel concentration, ranging from about 0.1 to 1.5 micrometers per second, as can be seen in the second graph. This observation of increased speed upon increasing fuel concentration was quite expected. However, it was a necessary first step prior to introducing depletion attractions, which, as you might suspect, could add a level of complexity to the suspension. As for the large error bars displayed, the origin of error has different sources, including particle fabrication and the nature of the experiments. For example, the platinum cap along the surface of polystyrene spheres may not necessarily cover 50% due to particles overlapping. Also, although we're working with a monodispersed suspension, variations in particle sizes are bound to be present, and that affects the propulsion speed as well. Moving forward, our second step involved introducing 20 nanometers charged nanoparticles to utilize depletion as a method for propulsion control. And for clarification, depletion force is an attractive force that arises between large colloidal particles that are suspended in a dilute solution of depletants, which are smaller solutes that are typically excluded from the vicinity of the larger particles. So upon initial observations, we noted that the charged nanoparticles did cause a decrease in the propulsion speed, as can be seen in the videos, which are sped up and on the same timescale. And furthermore, we conducted quantitative analysis to support our observations. By analyzing our experimental data via evaluation of the MSD graphs for active particles and 5% hydrogen peroxide with and without depletants, we concluded that increasing the depletant volume fraction caused the apparent swim speed to decrease. This can be observed from the two slopes, for which is the active no depletants and active with depletants. And the slopes, as you may recall from earlier, are the diffusion coefficients of the particles. So in other words, a steeper slope implies slower diffusion coefficient, which means lower propulsion speed, which is the case for active particles in the presence of depletants. This led us to hypothesize that depletion quenches the propulsion of active particles because it brings the particles closer to the boundary, decreasing the separation distance between particle and wall. And to support our hypothesis, we ran simulations from the separation distance inferred from experiments, and what we observed is that the magnitude of speed reduction was much smaller for experimental data when compared with the simulations. This suggested that in addition to hydrodynamic quenching, there's another parameter contributing to the quenching of particles. This parameter was concluded to be the conductivity of the suspension, which increases as a consequence of adding negatively charged nanoparticles. Additionally, we conducted supplementary experiments in order to separate out the effects of conductivity and hydrodynamic quenching on the particle speed. And we observed that the addition of polyethylene glycol, or PEG particles, with low conductivity, so less than 10 microsiemens per centimeter, resulted in decreased speed. This is seen in the first graph on the left, where the y-axis represents the propulsion speed in units of micrometer per second, and the x represents depletion volume fraction. And similar experiments were conducted using various concentrations of sodium chloride, thus varying the conductivity of the suspension, which ranged between the conductivity of ultra-pure water and about 80 microsiemens per centimeter, which goes far beyond the conductivity generated by the 20 nanometer charged nanoparticles. Thus, These experiments supported our observation that both uncharged depletants and the increase in conductivity will quench the propulsion of catalytic active particles. And this concludes all of our finding regarding particle dynamics at infinite dilution. Okay, so considering that our active particles are used as model systems, the next logical step required deviating from infinite dilutions and increasing the particle concentration, which allowed for analysis of particle clustering. And this segment represents our most recent paper. So a dilute suspension of active swimmers can form clusters even in the presence of repulsive forces. And the reason behind this phenomena could be explained by the collision dynamics of particles. So when two particles collide head-on, they block each other, forming a temporary two-particle cluster. And over time, particles will reorient themselves away from each other and eventually swim apart on a timescale of the rotational diffusion. However, in a low to intermediate particle concentration regime, and depending on the particle speed, the mean collision time could be fast enough that another particle joins the cluster before the two particles break apart, building larger clusters that could rearrange and exchange particles, forming this phenomena, which was first identified in the year 2013, and it's known as living crystals. And before I highlight our findings regarding particle clustering, I would like for you to note that the term intermediates refers to doublets and triplets, while the term cluster refers to aggregations of at least 4 particles. So based on our previous work, we expected the cluster formation to increase with increased nominal speed, and varying the speed as seen earlier was done by increasing hydrogen peroxide concentration. Also, the population count in these experiments were converted to probabilities by dividing each population count by the total number of observations in each frame. This was done to allow for a better comparison between different trials where the total number of observations varied. The figures above, they represent the evolution of singlets, intermediates, and clusters in 3% hydrogen peroxide as a function of time. And as can be seen, the probability of clusters observed increased from beginning to the end of the experimental observation at the expense of singlets and intermediates. This is not surprising, considering that it's far more likely for a cluster observation of 4 particles or more to keep its cluster classification after losing or gaining another particle than it is for intermediates. Next, the clustering dynamics were analyzed with varying hydrogen peroxide concentration between 0 and 3%. And as we can see, the nominal speed was effectively increased with increasing hydrogen peroxide concentration, which in return increased the probability of larger formations observed, as evident by the increase in intermediates and clusters, while the singlets declined. So in other words, an increase in peroxide concentration caused an increase in speed and collision probability, thus enhanced the cluster formation. Lastly, we studied the cluster formation as a function of depletant volume fraction for systems fixed at peroxide concentration of 3%, which are seen represented as circles, and the 0% peroxide, which is represented by the triangles. As can be seen from this graph, clustering initially was slightly enhanced at small depletant concentration in the presence of fuel, but was diminished as depletant concentration grew. So at low depletant volume fraction, the apparent speed of the active particles is roughly unchanged, meaning no significant change to clustering probability. However, once the particles collide and cluster, there's likely an enhanced attraction experienced by the particles due to presence of depletants. And at high depletant volume fractions, particles' propulsion speeds are reduced, thus decrease the collision probability and the extent of clustering. As for the Janus particles without fuel, they experienced low levels of clustering that were slightly enhanced from that of the Janus particles in the absence of both fuel and depletants. Note, the shaded area in the graph represents the region of volume fractions where the propulsion speed was observed to be quenched, which is also in agreement with our previous findings at infinite dilutions. And this concludes my presentation. Thank you very much for listening.